Thanks, Andrew. And it's great to see so many old friends and current friends gathered together in the same room. By the way, I'm customer number 1. So, I'm going to carry on the discussion, really, of the development of some of these interesting new contrast strategies, especially for applications with photoacoustic imaging. And I think you get the general idea that where we're going is the attempt to develop nano-sized particles that can report back information, not just about the vasculature, but what lies beyond the vasculature, and in particular, surface markers and other things that might exist in disease situations such as cancer. I have many collaborators, some of whom are listed here, and I do want to make sure that I give them credit for much of the work that's actually being done. So I, in particular, want to mention Gang Zheng and his lab downtown, and you just heard a talk on the porphosomes, and I'm going to stay away from the porphosome area, but I think that's really exciting work that's being done. My new colleague, Christine Demore, who's working with me over at Sunnybrook now, trying to advance the development of transducers and all of these interesting nanoparticle contrast agents. John Valiant, who's at McMaster University, who's a wizard with chemistry, so that when we have an idea that we'd like to put some kind of surface ligand on a particle, John is the guy who helps us do that kind of stuff. And Bob Prudhomme at Princeton University for some of the agents I'm going to talk about. Mikhail Shapiro at Caltech. And of course, Fujifilm and the team here who've been involved in the development of photoacoustics. So the lab at Sunnybrook is fairly large and fairly diverse. I'd just like to point out a few of my senior colleagues. So Christine is right here. Peter Burns, Joanne LaFloc, whose work I will describe in this short talk, and a range of our students and co-ops who come in for any period of time from 4 months to 8 years to do their PhDs, and who contribute to all of these projects. So in this talk, I'm going to first of all give you an overview of nanoparticle photoacoustic contrast agents, and then I'm going to talk about this interesting problem of trying to break down the blood-brain barrier so that we can deliver some of these particles into the brain tissue itself. And I'll then talk about the development of some really novel agents which are genetically programmable, but offer us the potential to combine photoacoustics and ultrasound detection in the same package. And finally, a more translational topic, prostate photoacoustics, which is something that we're actively working on. So what are the possible methods that we could use to develop photoacoustic contrast agents in the nano range? A lot of work initially was done on plasmonic nanoparticles, and this field is very large, very diverse, and of course focuses on metallic structures such as gold nanotubes and nanocages and things like that. And then more recently we've worked on the development of dye particles, where we take dyes and instead of just injecting them as you might with ICG or something like that, we try to encapsulate them into nano-sized packages and then inject those packages. And that's work that we've been doing with Bob Prudhomme. And also looking at dye-labeled protein nanostructures. So if we can create some nanostructure and then label it with the dye and send it in as a molecular imaging agent, that's also of interest to us. And this is where we get into the genetically programmable type particles and the methods that we can use to actually attach these things to useful substrates that we can inject into the mouse. So let me talk about the dyes first. So one of the problems with dyes are that they're the ones that are water soluble, such as ICG, rapidly bind to serum proteins, and they do tend to get cleared rather quickly from the tissues that we're actually interested in. Also, the ones that are hydrophobic contain these large aromatic structures which are not really very soluble, so they're very difficult to get into solution, very difficult to get them into some kind of packaging that allows them to be observed inside the tissue. So the motivation is to overcome that problem using some interesting chemistry that was developed at Princeton. And the basis of that chemistry is something called flash nanoprecipitation. So we start with those highly hydrophobic optical dyes, and we repackage them so that they become biocompatible and water dispersible, with size ranges from about 40 nanometers to 250 nanometers. And then we can also go through the chemical process of altering the surface, putting PEG on the surface, making them less susceptible to the immune system. And this work was described in a number of papers that have come from Bob Prudhomme's lab actually over the past 20 years. So it's been a long process of development. So basically how it works is this. Take a number of streams which contain, first of all, the organic core, so that would be the dye, put on a PEG stabilizer and a fluorescent marker, if you'd like to do that, run them through this rapid mixing chamber, and through that process create these particles where the the dye particles are confined to the core, and then you have these PEGs which can then be labeled with the fluorescent markers. So now it's bi-labeled. We have the fluorescent part of the particle that we can detect optically, and we have the core particle that we hopefully can detect with photoacoustics. And just to give you an example of how well they can tailor the absorption characteristics, here are 3 particles that are shown. We're looking at absorption versus wavelength here, and you can see how sharp these resonances are. So now we can use unmixing to detect all 3 of these after a single injection. So we can multiplex this whole process using a number of different dye particles. And just as an example, probably can't see this because of the lighting here, but in this case, these particles have been incubated with some of these dye nanoparticles, and you can see in these breast tumor cells the accumulation of the dye, which is shown in green, surrounding the red nuclei, which are labeled with a different marker. So clearly they can get into the cells, they can penetrate the membranes, and they can become our beacons for us to detect photoacoustically. So now I want to come back to this problem, which is really how are we going to, because one of our big objectives is to get these particles into the brain, because we can also conjugate and create particles that not only have dyes in their core, but also have drugs. So we want the drugs and the dye to go in and report their location in the brain, but we have to overcome the blood-brain barrier. So that brings us to another interesting experiment, which involves the use of both low-frequency ultrasound to disrupt the blood-brain barrier and high-frequency photoacoustics to detect how these particles distribute themselves in the brain. So here are our 3 dye core particles, which are shown here, the absorption spectra repeated from the previous slide. And just want you to note that the nanoparticle size is quite different, starting at 40 nanometers right up to 240 nanometers. Each one is labeled with a separate Alexa fluor, so we can track them fluorescently. And they have a particular name which is a little bit difficult to interpret, but essentially think of them as a size range from 40 to 250 nanometers injected into the mice that we're going to examine. So here's the experiment. It consists of, starting at time t is equal to 0, we disrupt the blood-brain barrier. So here we're using low-frequency ultrasound focused into the mouse brain, and we're going to open the blood-brain barrier and using conditions that are described in detail here. I don't want to go through these. They're well described in the literature. It involves the injection of microbubbles, and the low-frequency ultrasound is used to localize a region of the brain where those bubbles will allow smaller particles to diffuse. How do we check that? Well, we put the mouse in an MR scanner, and we actually look at gadolinium diffusion after we've opened the blood-brain barrier. So you can see 4 regions here where the blood-brain barrier is open. So then we can start doing our photoacoustic imaging experiments. And what we do is we inject a cocktail that has all 3 of those agents in the cocktail and inject them all together. And once we've done the photoacoustic experiment, we'll spectrally unmix them and decompose them into these regions. But here you can see a 3D picture following the blood-brain barrier disruption, showing the photoacoustic signals lining up quite well with what we saw in the experiment that we did with magnetic resonance imaging. And our colleagues would dearly love to be able to not have to do these MR experiments. Number one, because it takes so long and it's so difficult to perform. If there was an easy way to do this in vivo, this would be the way to go. So I just want to show you some of the images that have come out of this experiment. So we're using, as I said, a variety of nanoparticles, starting from 40 nanometers up to 240 nanometers. And in the experiment where we're working with the ex vivo brain, so this is excised and perfused, so there's no blood to confuse the situation, you can see clearly the spectral unmixing detecting all four of these, or all three of these particles quite effectively. When the brain is perfused, but the skull is still intact, it's less clear, obviously, because we're going through the skull. But you can still see regions of the brain where the blood-brain barrier has been disrupted here. And if we do it in vivo, again, there's more noise because of the influence of the skull, but we can, in certain situations, detect the disruption in the brain. And we can confirm that by then slicing the brain and looking at the fluorescence images. So we can confirm that the regions that we thought were opened have in fact been opened. In this image, we're just looking at three-dimensional views where we project through all of the different planes looking down on the brain, and you can see the regions where the blood-brain barrier has been disrupted quite clearly here. And you can look at these things in the transverse view as well. So here we're cutting the brain in the transverse view, and you can see all three of these regions lighting up. And what it shows, and what the most important thing it shows, is that these nanoparticles are actually leaking out of the vasculature in the brain and entering the brain tissue itself. So the possibility of using them in drug studies is a very intriguing thought. So I wanted to move on from blood-brain barrier to talk about another area of interest of ours, and this goes to talk about the same sort of particles that Mike Koleos was talking about earlier. So he talked about these nanobubbles that he used in his studies, and we have been working with our colleagues at Caltech to develop protein nanostructures which might also be used in contrast experiments and in photoacoustic experiments. So these particles are actually were developed in nature by bacteria and used as a flotation device. So there's a series of genes that actually code for small protein nanostructures called gas vesicles that are used by bacteria for flotation. So the bacteria use these particles to move around in the medium in which they're living. And here's a vast lake of halobacteria growing in Bonaire, and we look at this as a vast lake of contrast because we grow these particles in our lab now. We harvest them. We actually harvest those gas vesicles and use them as contrast agents. And so what we're exploring now, what would the applications of such agents be? And of course, our greatest interest would be in making sure that they can get past the vascular barrier and into the tissues that we're most interested in. So just to give you an idea of what these things look like, here are some EM images of Anabina, which is a bacteria, and halobacterium, which is shown over here. These are the actual gas vesicles that are made out of a protein, and they're filled with gas, and they're naturally filled with gas. They self-inflate, and they create good acoustic scatterers the same way that Mike Koleos's nanobubbles are strong ultrasound scatterers, and they're equally nonlinear. Now the exciting thing about these particles is that they can be genetically programmed, which means that eventually we may be able to program these into eukaryotic cells and get them into systems and be able to track stem cells and things like this on the basis of the production of these particles. At any rate, they're roughly anywhere from about 50 to 1,000 nanometers in their dimensions, and they're genetically relatively uniform, so they're quite easy to work with. And there's been a lot of work that's come out of our interactions with the Shapiro lab over the past few years. And we're discovering a lot more about these particles every day. So we started with a paper in 2014 in Nature Nanotechnology, where we described the potential of these particles for imaging, and a bunch of papers discussing how the physics of how these particles work and how they're produced and how you can produce them. One of the things I would like to get to accelerate would be for other people to start using these nanoparticles in some of their experiments, whether or not they're contrast experiments or photoacoustic experiments. There's just a wealth of interesting things to look at here, and we'd like to get more people interested in that. So we did publish a paper, like a 50-page paper, a protocol for exactly how to make them. And they're not that difficult to make. You could go home to your lab and start making these things relatively simply, and if you have any problems, you just call us and we'll tell you how to do it. But I'm just saying, there's a lot of great work to be done here. And the most recent paper coming out of the Shapiro group at Caltech made the front cover of Nature, which is no small feat, I can tell you. And it described, I think, cellular sonar. I don't know if you can read that up there. But this was the first acoustic reporter gene reported in the literature. And so what Mikhail did is he took these particles and he altered them, and he got them expressed in E. coli. And E. coli, as you know, colonized the gut. And he was able to detect the particles in the gut and demonstrate the fact that he could make them grow and report on their activity as they circulated through the body. So we're not quite into eukaryotic cells, but we're getting awfully close. I just want to show you a few of those experiments. So here we are with 2018, and we now have a demonstration of acoustic reporter genes. And these are the bacteria that are shown here. And in order for them to make these bacteria visible, they had to actually combine the genetic characteristics of one bacteria with another bacteria. So they took some genes from a bacteria called Megatherium and the Anabina floss that I described earlier, and they used each of the genes that actually create these particles. There are 11 genes or 12 genes. 3 of them are really important, A, B, and C. So they changed those around and connected the two. So you now have Anabina for A, B, and C, and Megatherium for the rest of the genes in this cluster. And when he created those particles and expressed them in E. coli, all of a sudden he was able to get good signals from these bacteria. And you can see how full they are with these nanoparticles as they grow inside the bacteria. And down here you can see the increase in signal that he was able to show. And then in terms of doing this in vivo, he demonstrated it in mice. And I don't want to go through the details of the experiment, but here are some in vivo images where we're looking from the surface of the mouse here down into past the bladder and looking at the gut. And here you can see the regions of the gas vesicles as they're growing in the gut. So a traceable reporter gene for ultrasound. Now in understanding these particles a little bit better, and one of the things that we want to do is to decorate the surface with useful things that, that we can use in our experiments. And in order to do that, I've worked with John Valiant at McMaster, and we use an interesting chemical process called click chemistry, and that's briefly illustrated here. So we have a halobacterium, which is here, and we want to combine that with a structure here, which is called TCO, transcyclooctene. So you see the process here where that is now on the surface, and then we conjugate it with either a radionuclide so that we can use PET imaging to track where these particles go, or we can put on a dye on the surface of our gas vesicle and image them fluorescently and track where they go. So we did functionalize the gas vesicles and attach the, whoops, we're going backwards here. When we put the radioactive tracer on and do imaging, you can see with the technetium conjugation, we can now look at what the biodistribution of these particles are in the tissue, and we can show the translation of the particles through the gastrointestinal system and follow it as it's eliminated from the body. And we're very interested now in attaching agents that can be photoacoustically active as well. So this is an example of some studies of using this transcyclooctene tetrazine chemistry to do some interesting photoacoustic experiments as well. So what we're looking at is a spectrum of photoacoustic signal versus wavelength in this direction, and we're using that phantom that Andrew showed earlier, where each of the channels has a different material inside it, and we're looking at how photoacoustically active they are. If you look at the spectra, you can see some interesting things here, and I'm just going to focus attention really on these two lines. The first one, which I believe is S3, this one right here, is a combination of the dye that we're trying to conjugate with the particle and with a chemical called bisphosphonate. And the reason we wanted to combine it with bisphosphonate is that we wanted to use this as a tracer that would go to bone metastasis, and bisphosphonate is deeply involved in that process. So they're busy at McMaster doing these experiments right now, and I understand they've been reasonably successful, and I'm hoping at future meetings they'll report on that. But you can see the very strong signal coming from that particular conjugation of that molecule, which now can be used to trace metastatic development in bones, and you get a very strong signal to noise here, just above 800 nanometers. The other line that's shown here is the signal that comes from the conjugation with gas vesicles. So we've turned our gas vesicles into photoacoustic agents as well, and this also gives a very strong response. So we're very anxious now to test those in some of our future studies in the development of these agents. Now I just want to finish off with just a very practical description of how we see this evolving to develop a translational photoacoustic system for working in the prostate, just to follow the work that was reported this morning. So we're going to work with a system which is developed by Exact Imaging that's shown here for prostate imaging. I'm going to quickly go through some of these. Just to show you what the images from this system looks like, you can see really a great high degree of detail in the prostate, including, in this case, numerous microcalcifications. But I'm sure you can appreciate the high level of resolution that this 20 megahertz prostate probe gives, and what we want to do is turn this into a photoacoustic probe. And the plan to do that is to use the transrectal ultrasound probe developed by Exact Imaging, which has the ability to do biopsies in real time. And we're going to add some channels or fiber along the array itself to push the optical energy into the tissue from the rectal side. And we're also going to combine it with a transurethral optical fiber to illuminate the prostate from the inside. And we've just started doing ex vivo experiments, and some examples of those experiments are shown down here, where we're looking at the capsule of the prostate, but this is ex vivo. And unfortunately, of course, in this situation, the blood is largely drained out of the tissue, so it's not particularly informative. But we can show at least that the combination of the two, transurethral and transrectal, gives us photoacoustic signals on both sides, and we're going to try to balance those out to get more effective photoacoustic imaging of the entire prostate. And this is one of the first images that we made with the laser X system in a canine study in a live dog. And this is the prostate in this region. This is a transurethral catheter that's sitting in the prostate. You can easily see the catheter and the intervening tissue. And fortunately for us, the most important part of the tissue lies between the urethra and the rectum. So we're hoping we'll be able to get similarly good performance when we finally do this experiment in vivo. So I just want to conclude by saying that both endogenous and exogenous photoacoustic contrast agents offer valuable imaging biomarkers for the functional assessment of healthy and diseased tissue. These nanoparticle contrast agents that are based on a wide variety of constructs are going to be vitally important to the future of the field. In fact, I think they are the most important future for photoacoustic imaging. And it is possible to disrupt the blood-brain barrier to reveal the extravasation of photoacoustically active contrast agents that have been created using a variety of approaches. So we're hoping to move those forward and do studies of disease models and drug interventions. So with that, I'd like to conclude and say thanks.